Welcome back. And we are going to start with chapter 4, which is the archaeal cell structure. It's a fairly short chapter. And then we are going to compare the differences between the bacterial cells, the archaeal cells, and the eukaryotic cell. The next chapter, which is chapter 5, is the eukaryotic cell structure. But we have covered the whole chapter pretty much in a different class, like a cell biology. And that's why we are not going to cover it. Just the comparison is actually at the end of this same video. So we're going to take a look. So we are going to describe a typical archaeal cell and discuss the key differences between the bacteria and the archaea. So one of the common features with bacteria is the genes for metabolism are pretty similar. On the other hand, for the uniqueness of the archaea, they have we talked about that this is the basis that they were actually put in a different phylogenetic category, is the ribosomal RNA gene sequence. Now, the ribosomal structures is all very close to the bacterial ribosome, but the sequence is very different, and that was the basis of classification by Carl Woese-Fox that we talked about in chapter 1. And many of these bacteria are actually capable of methanogenesis. So they actually do produce these greenhouse gases, and a lot of these archaea are actually living in different manure and all those kind of rotten material that actually produce these methane gas as such. Highly diverse in respect to morphology, physiology, reproduction, ecology. They are found in very varied conditions, many a times in extreme conditions. We are going to see that. And they are best known for growth in anaerobic condition, hypersaline, that means a very high salt concentration and pH extreme, and high temperature habitats. So this is an interesting part that they probably evolved and during the very early stages of life, even before the earth actually got into the oxygenated form. Due to photosynthesis, the archaea actually thrived way before that, when the conditions on the earth was actually very, very harsh. So we will discuss the comparison in the chapter 5, which will be just at the end of this lecture video. So let's first take a look at the archaeal shape, arrangement, and the size. Much like the bacteria, cocci and rods are actually common shape. Other shapes do exist, but there is no spirochetes or spirillum or mycelial forms, or branched or flat shapes like actinomyces are also not available, not found. The sizes also vary, sometimes about 1 to 2 micron to 1 to 5 micron rods, and 1 to 3 micron diameter. So you can see the size are actually pretty close to the bacterial size, and the smallest is about 0.2 to 0.4 micrometer. And we have seen that average size of the mycoplasma, which is also in a bacteria, doesn't have a rigid cell wall, is about 0.3 micrometer. So they are pretty close in that. Largest so far is about 30 millimeter in length. So you can actually see that this is a multicellular form, and this is definitely visible by the naked eye, but again, not a single cellular entity or structure. So what you are looking at, that on the left, you are looking at this Methanosarcina mazie, a coccus from clusters such as viewed by a scanning electron microscope. You can see that. On the right here, you are seeing an electron micrograph, which is a transmission electron micrograph. And this is a bacteria called Thermoproteus tenax, and it's a branched archaeal cell. So this branched archaeal cell, you can see that it's branched right here. And on the right here, you can see that these filaments are actually covered by a biofilm in rod-shaped bacteria. And these bacteria may actually be symbionts of host archaeans. So they can attach to the host by this filamentous structure, and this forms a biofilm. So these are some of the structures, and you can see that if you look at this overview of the structure, that may look like pretty much the coccus, like the bacterial structure, but they are actually different. So let's take a look at the bacterial cell envelope. So in the cell envelope, it's differ from the bacterial envelopes of the molecular makeup and organization. And the S layer may be the only component outside the plasma membrane. And that is the most important part, that they do not have any peptidoglycan layer by the bacterial cell wall. And some of them lack cell wall completely, just do have a plasma membrane and maybe an outer membrane, and that's all they actually have, no cell wall at all. And the capsules or slime layers are also present, but they are relatively rare compared to the bacteria. So what is the difference of the archaeal membrane? So the archaeal membrane are not made up of phospholipid bilayer. So here we are actually looking at a typical phospholipid bilayer that you can actually see in the bacteria. You can see in the eukarya as well. So here on the top layer, you have the head group, phosphate, and two fatty acid tails. And towards the cytoplasmic surface, you have the phosphate head group, two fatty acid tails. These are some transmembrane proteins. So this top one is an outer leaflet, and here you have an inner leaflet. So this is a phospholipid bilayer. On the other hand, what you actually see then in archaea, they are actually made up of a monolayer. It's a single layer, but it acts as a bilayer. So how this actually happens? So the first thing that what they are made up of, that they actually are made up of isoprene units, and these isoprene units are actually 5-carbon branched structure. You can look at the structure in your textbook, and due to this isoprene unit, the lipids actually pack in a different way, and it has to do with that how they can actually withstand that extreme environment or temperature is due to these isoprene 5-carbon branched structures. The second thing that what they have, that in here, what you are seeing in the phospholipid, you have a phosphate head group, and these fatty acids are actually attached by a glycerol molecule, and this glycerol actually do have an ester linkage with this. So this is in the phospholipid, you have this ester linkage. So ester linkage is common in eukaryotic plasma membrane and as well as bacterial plasma membrane, whereas in archaea, what you see that instead of a ester linkage, you actually find an ether linkage. And this is a very, very different as such. So what you are seeing that these are a closed molecule. So what you are seeing right here, that this is a bilayer, and there are about 20 carbons in the fatty acids. So it runs from here, 20 carbon, runs from here, 20 carbon. On the other side, you may have 20 carbon as well. So total in the length is probably about 40, just roughly, and these numbers actually do vary. On the other hand, for the archaeal membrane, which is just a single monolayer, this head group is attached with this 40 carbon chain, and you have another head group on the other side as well. So you have two head groups, and it's a closed molecule. So this acts as a lipid bilayer, but this whole molecule is a single molecule. We'll take a look at them in a detail in a second. So they differ from the bacteria eukarya in having the branched chain hydrocarbons attached to the glycerol by ether linkage. So remember that in the bacteria eukarya, they are ester linkage. Here they are ether linkage. And they actually have polar phospholipids, sulfolipids, glycolipids, and unique lipids are also found in the membranes. So let's take a look at what we are looking here. So here, this is a typical isoprene, which is a branched hydrocarbon, 5-carbon hydrocarbon, and then it is attached right here with a glycerol molecule. And you can see that the glycerol molecule will be attached to the fatty acid, and this is an ether linkage right here. This is the ether linkage. This is a head group, which is polar. This will either be towards the environment, which is the outer leaflet side, or the head group will be towards the cytoplasmic, which will be the aqueous phase as well. Because the head group has to be polar so that it can actually interact with the water towards the environment and also the aqueous environment in the cytoplasm. So here you can see that you actually do have a head group. Then with the ether linkage, you have the fatty acid right here. The fatty acid is continuing as a 40 carbon, and these fatty acids are attached to another ether linkage to another head group on the side. Sometimes you can have this cyclic, these molecules actually undergo cyclization. So these are the cyclic molecules that are actually formed in some of the archaea as well. There are some rare types, but there are some where you have the lipids where they are actually not closed. So you have a smaller number of carbon lipids, which is just like a phospholipid bilayer. But these are these are also not very common. You have many other types as well, but these are somewhere there as well. So on the other hand, what you are seeing that this side is the bacterial membrane or the eukaryotic membrane, and right here is the polar head group, and this is the glycerol molecule. And the glycerol molecule is attached to the fatty acid right here. This is one fatty acid here. There is another fatty acid here. And this bond right here is an ester linkage. So this is the important one. This is an ester linkage. This is an ester linkage. Here, the linkage is ether linkage. This is an ether linkage. This is an ether linkage. So that's the basic difference as such in between the archaeal membrane as well as the bacterial membrane. And you can see that usually the carbon number in the fatty acids are about 20. So it runs on this direction, and these are just one phospholipid molecule. On the other hand side, you have another lipid molecule which is coming from the other. So you have two leaflets, an outer leaflet with one head group and inner leaflet with another head group with the C20, or the number of carbons are 20. Here you have 40 carbon with two head group attached, and it's a single molecule. It's not, there is no two leaflets. There are just some examples of these types of molecules are also present, but these unique molecules actually are more abundant. So these are also called these diglycerol tetraethers. There are some diglycerol diethers. So diglycerol diethers means that you have 1 and 2 diglycerol diether, this one end, diglycerol diether, diglycerol tetra, 1, 2, and 3, 4. So these tetraethers are like this, which are the closed circuit molecules right here. You can see that right here, O and O. So this is an ether linkage 1, and this is another ether linkage 1. So 1 and 2. So this is at one end. This is 1 and 2. So right here, if you can see that these are the two OO, these are the two ether linkage in this molecule. So this molecule is a diglycerol diether. This molecule is a diglycerol diether. On the other hand, this molecule and this molecule, you can see 1, 2, and then other side also 1, 2. This is a diglycerol tetraether. This is a diglycerol tetraether. These two closed molecules. So the advantage is that the diglycerol tetraethers can actually withstand more extreme environment compared to the diglycerol diethers, which are similar or closer to the ester. Again, remember that these are ether linkages. These are ester linkages right here. You have ester linkages here. So the ester linkages actually cannot withstand so much of the environmental harsh condition. So this is one of the very, very unique difference in between the two types of membranes. On the other hand, the cell envelope is a varied S layer attached to the plasma membrane, and sometimes this S layer is the only layer attached to that, and which could be up to 70 nanometer in width. And there is a structure which we call as a pseudomurine. This is a peptidoglycan-like polymer, but it is not peptidoglycan. There is no real peptidoglycan present, and we are going to see that. And there are complex polysaccharide proteins and glycoproteins are also found in there. And this is a species which is the Ignicoccus, and in this one you have only one outer membrane. There is nothing rather than the outer membrane that is present in this. So very, very different from the bacteria. So you can see that the archaeal cells, many of them use the same mechanism of the nutrient uptake and exhibit in the bacteria, like a facilitated diffusion, active transport, primary, secondary. And they also use the phosphoenol pyruvate sugar phosphotransferase system group translocation mechanism that's found in some of the archaea. So these are some of the archaeal cell energetics and nutrient uptake as well. Just a very rough idea. On the other hand, the bacterial archaeal cell wall, actually not bacterial, lack peptidoglycan that we constantly saying. Most cell wall actually do have the S layer, and many protein sheath external to the S layer as well. And the S layer may be outside the membrane and separated by this pseudomurine, or this is a pseudopeptidoglycan, which is not a real peptidoglycan. We are going to look at the structure soon. And pseudomurine sometimes could be the outermost layer, similar to gram-positive organism, and sometimes there could be a slime or capsule layer outside that as well. There are many different types of variation that we are going to take a look in a second. So here, what you are seeing that in some archaea, the S layer is the outermost layer, and separated by the plasma membrane by a peptidoglycan-like molecule called pseudomurine. And pseudomurine actually differs from the peptidoglycan in that they actually have this L amino acid, L isomers of amino acid that is present in eukaryotes. You can see that there were some L amino acid like lysine was used in the bacterial peptidoglycan, but many of them were actually D isomers of the amino acids. So this is a drastic difference from the other. And what you can see that the D amino acids, on the other hand, the L amino acid instead of D amino acids. And another important difference, what you actually see, that the two sugars that you have in bacteria, they were N-acetylmuramic acid. So instead of N-acetylmuramic acid, this is called this N-acetyl allosamuranic acid. So N-acetyl allosamuranic acid and N-acetylglucosamine is actually the same. So instead of muramic acid, it has been changed by another different sugar. And then right here, you can see that right here, it is written as beta 1,3. So right here, the bond in between that N-acetyl allosamuranic acid, on the other hand, N-acetylglucosamine, this bond is a beta glycosidic bond in between 1 and 3. 1 and 3 beta glycosidic bond. Whereas in the bacteria, this will be a beta glycosidic 1,4 bond. So the first carbon is attached to the fourth one. Here it is, the first carbon is attached to the third one. So these are the most important part, is beta 1,3 instead of beta 1,4. On the other hand, you can see right here that the S layer of the plasma membrane, the cytoplasm, this one is actually, you can see the cytoplasm plus plasma membrane, then you have an S layer. So these are found in the Methanococcus, Halobacterium, those kind of bacteria. Again, you do not have to memorize each and every one of them, but you can just appreciate that how much differences are in here. On the other hand, here you can see there is a cytoplasm, plasma membrane, S layer. You can have a protein sheath outside right here. And these are actually found in the Methanospirillum type of bacteria. Here you are seeing that the cytoplasm, plasma membrane, S layer, and then you have a methanochondroitin right here, another layer. And this is basically, it is a glycan in this methanochondroitin, and the methanothermus, methanopyrus, these are all actually methanogenic bacteria producing methanogens as well. Here, on the other hand, you can see the cytoplasm, plasma membrane. Then you have this pseudomurine layer. There were no pseudomurine there. Here, this is the pseudomurine. You have this layer, and then you have a S layer as well. So this this type of, on the other hand, is actually available in particular like methanopyrus, methanothermus. Those are the bacteria which actually does have these type of arrangement. On the other hand, you can see that you have a polysaccharide. This is in the, like a capsule or slime layer outside the plasma membrane, and you can just only have that. That's it. There is nothing else. So this type actually can be present in some of these methanothermobacter, then methanosphaeria, those kind of bacteria. This is another glycocalyx, which is actually loosely arranged. So the difference in between that, this is a polysaccharide right here. This is more like a capsule because it is tightly arranged. Whereas here you can see this glycocalyx is a little bit looser, but this is loosely arranged on the just compared to a tighter polysaccharide. But glycocalyx is also the same way as the slime layer. And these type of actually find in a thermoplasma species. On the other hand, there is the last type right here where you have the plasma membrane, then you have an intermediate compartment and then you have an outer membrane as well. And the Ichthyococcus that we just talked about actually also in very, very harsh environment, they actually have this double layer membrane structure as well. So these are some of the differences in the cellular structure that you can actually encounter in the archaeal cell wall. So let's take a look at the cytoplasm, very similar to the bacterial cytoplasm and lack the membrane-bound enclosed organelles. Many contain inclusion bodies, for example, gas vesicle for buoyancy control, and they also do have similar type of structures. Also, usual ribosome, nucleoid region, and some structures may be different. But however, the complex structures are like the ribosomes, sites for protein synthesis, actually is to some extent similar, but they are very different in some way. So the entire ribosome, if you just look at the bacterial and the archaeal ribosome, they are 70S, 75th Svedberg unit. And eukaryotic ribosome, on the other hand, is 80S. So the bacterial archaeal ribosome, if we just separate the small and the large subunit, the small subunit is made up of 16S ribosomal RNA and the 23S and a 5S large subunits. Now, what happens that although the sizes of the ribosomal RNA are the same, but their sequence actually distinctly varies, which is the basis of putting them in a different domain. And at least one archaeon have an additional 5.8S rRNA, also seen in eukaryotic subunit. So here, 23S, 5.5S, and you can have an additional 5.8S as well. Proteins of the ribosome actually can vary as well. So archaea are more similar to eukarya and actually can contain 30 or 40 more proteins, and they are very unique to the archaea as well. These proteins are associated with this ribosomal RNA to give them two subunit structures. So remember that to make the 70S structure, you have the two subunit, and they are 30S and 50S. 30S and 50S make the 70S. Here, the 40S and 60S make the 80S. So think about that this is the 70S, the small one is 30S. So the Svedberg unit of 16 is contributed by the ribosomal RNA. The rest of the Svedberg unit, which actually makes up to the 30S of the smaller subunit, comes from the protein weight. And the same way that 23S and 5S altogether is about 28S, if you do not have that one, then the rest of them, which actually make to the 50S, which is almost 22S, that weight is given from the protein in the classification of the size of these ribosomes. So the nuclear region, on the other hand, is irregularly shaped in bacteria archaea. Usually it's not a membrane bound. A few exceptions, which are usually the invagination of the plasma membrane, but it is not completely enclosed nucleus like nuclear membrane, which is separate from the outer membrane. And usually there is a single circular chromosome associated proteins. And there are some evidence of polyploidy in some archaeons. That means that you can have multiple chromosomes in the same structure, just like the bacteria as well. And the supercoiling and nuclear associated proteins actually contain histones. So this is a very big difference from the bacteria. The bacteria actually do supercoiling, but there is no histone present in there. But the eukarya actually contain histones, whereas the archaeon also contain histone. But there is a difference in the nucleosome formation when you have these wrapped around nucleic acids. In the eukaryotes, you actually do have 8 histones molecules or 8 nucleosomes, where the nucleosomes number of this, where you have these histones, are 4, half the number of the eukaryotic nucleosomes. So the structure of the histones in the nucleosomes are different. It is 8 histones in the eukaryote, whereas 4 histones in the archaea. So that's the big difference. But histone is not at all present in the bacteria, but it's present in archaea. So also we can take a look at the external structure. So first of all is pili, and it's not well understood as of yet. And some of the composed of the pilin protein of homologous bacterial type 4 pili proteins. Type 4 pili proteins are essentially the outside of the cell surface pili. These are called the type 4 pili or TI4. So they may be involved in adhesion mechanisms. There are some other structures have been seen, which is not seen in a bacteria. These are called cannulae. It's a hollow tube-like structure on the surface of this bacteria, which is a thermophilic archaea in the genus of Pyrodictium. And this cannulae, actually, the function is actually not known. It's maybe involved in the formation of network of multiple daughter cells, and some kind of interaction in between the cells may be carried out by the cannulae, but really the scientists still do not know exactly what is the function of the cannulae. But you can see that structure right here. So on the other hand, the hemi is not also well understood. There are 2 different types. What you are seeing that right here in this electron micrograph, you have an archaea and probably hundreds of these coccus-shaped archaea, And these hundreds of different hemi are actually radiating out from this archaea. So this you can see right here. On the other hand, this is the structure which we call as a grappling hook. And this is the end of one hemi. So there's multiple of them. If you just look at the ultrastructure of just one of them, you can see that there is a hook in there. So scientists actually argue that they may be involved in the adhesion mechanisms, but these some of the functions of these are still not well understood. So the next one is we are actually talking about the archaeal flagella, sometimes called the archaea. And the flagella is much, much thinner and much less complex compared to the bacteria. And there are more than one type of flagellin protein. You can see that the flagellin protein, which makes up the bacterial flagella right here, stacks up one upon each other. You have a different color here. You have a different color here. So you can see that they are made up of different flagellin proteins. But this is actually the same way that this is the filament. Then you have the hook structure right here. And then you have the basal body. And you can see that the basal body are very, very different compared to the bacteria. They actually do contain a lot of these flat group of protein. And this is actually more kind of related to this type 4 bacterial pili, which is made up of pyelin protein. And this pyelin protein is kind of similar to that. On the other hand, what you are seeing, this flap F, along with that, it's been shown that this is the plasma membrane, and right here is the S layer, and this flap F is actually responsible for anchoring the flagella onto the bacterial envelope. And this is the motor complex that we have seen, that these are actually made up of different flag J, flag H, flag C, D. There are multi-mers of proteins which actually function for the rotation or the propulsion of the rotation of the motor and the twitching motion of the flagella. But there is a very big difference in between the bacterial mechanism and the archaeal mechanism. In the bacteria, we already talked about that. Suppose this is the bacterial outer layer, and you have periplasmic space. And in the periplasmic space is high in proton, and as proton goes down from the periplasmic space to the cytoplasm, the proton motive force actually drives the flagellar motor. Here, there is no periplasmic space to have this gradient of the proton. So here, this motor is driven directly by the ATP. So all the food or nutrients that is actually absorbed by the archaeal cell, they actually made into ATP after the metabolism, and these motors are run by those ATPs as such. So here, another important thing that what you are seeing, that this is actually the flap B, which is the subunit of these structure. This is the flagellar structure. So compared to the bacterial flagella, where you actually have a hollow core, and how it happens that the component or the proteins which actually make up the flagella goes through the hollow core and added at the top. So this is how it grows over the top. On the other hand, here in the archaeal structure, these are the subunits of this flap B right here is a subunit of the flagella. They are added to the base. So not at the tip, but at the base. So very different in bacteria. New ones are added constantly on the top as it grows because it goes through the hollow tube. Here, the proteins are added at the base. So a major difference here. So we are still learning the fundamental basics of the structure function of the archaea, and that's the reason that this chapter is so small compared to the bacteria. And it's also very critical to understand the similarities between the archaea and the domain bacteria, and where the material yet to come on eukarya and similarities between those two. So let's take a look at the chapter 5, and we are going to only talk about the section 5.8 only, where we are going to compare the eukaryotic cell structure with the bacteria and the archaea, just in a nutshell. So here you can see that if we just compare some of the eukaryotic cells with the archaeal or the bacterial cells, the eukaryotic cells do have a nucleus, very different, and organelles, which are membrane bound. They are large, more complex. The archaea as well as bacteria are way smaller compared to an eukaryotic cell. They actually do have, the eukaryotic cell have meiosis, mitosis, much complex type cell division. And these are actually absent in the bacteria and the archaea, where they actually reproduce just by the binary fission. So one chromosome actually becomes just two. On the other hand, there are many of the biochemical processes and metabolic pathways are very similar, but also there are some difference in the shape, number, and the size of proteins, type of proteins. They actually vary, but overall, the processes actually do have much similarities, and the genetic codes are also the similar. Say for example, the codon which actually code for the amino acids actually are very, very similar. If AUG is a start codon in the bacteria, it is the same in the eukaryote. And if AUG is code for methionine, the amino acid, it will code the same in the bacteria. But there is some difference in the preference of the codons that the eukaryotic cells actually use, or the bacterial or the archaeal cells actually use. So there could be some usage differences. So here here is some overview of the structures. You can see a cell wall, a nucleoid region. Underneath underneath the cell wall you have a plasma membrane. On the other hand, if you just look at even a protozoal cell, you can see that how complex this structure is. And you can see that there is a membrane-bound nucleus. You have a nucleolus region, which makes the RNA. You have eukaryotic flagella. You have these vacuoles. These are similar, but you can see that you have, on the other hand, these thylakoids, which are the parts of the chloroplast. And these are membrane-bound organelles that you can see here. And there are some starch granules like inclusion bodies, and those are very similar. So if you can see that this is probably an euglena, which is a photosynthetic eukaryote, a single cellular protist, and it is a much, much more complex structure compared to a bacterial or archaeal cells. So let's take a tabular look at that. What are the differences in these? So you can see that we talked about that there is no membrane-bound nucleus in bacteria on the archaea, and DNA is not complexed with the bacteria with the histones, whereas some archaea, the DNA is complexed with the histone, but the number of histone per nucleosome is a little bit different, so it is really not exactly the same, but similar. So on the other hand, here you can see that in the bacteria, they usually one chromosome, circular chromosome, and the chromosomes have single origin of replication. Some are polyploid. One circular chromosome usually, some could be multiple origin of replication, some are polyploid, whereas almost always the eukaryotes have multiple linear chromosome with multiple origin of replication as well. So the reason that the eukaryotic genome is much more bigger, and if you do not actually have multiple origin of replication, then it will take forever for the chromosomes to replicate. Plasmids are very common in bacteria. In the archaea, in the eukaryotes, it's actually rare. It is present. Say, for example, the yeast cells. In the yeast, there are plasmids. They are eukaryotes, but it is much rare in higher organisms. So the introns are actually rare in bacteria and the archaea, where it's very common in the eukaryotes. What you mean by this, or in the DNA, you have the sequences where they code for the genes. Those are called the exons. And these exons are actually interspersed in between the introns, and the introns are actually non-coding DNA. So there is no non-coding DNA very rare in bacteria as well as in the archaea, whereas it is extremely common to have introns in the eukaryotes. And actually, the interesting part is that out of our whole genome, there is only 1.5% to 2% of our total DNA that you have in a cell. Only 1.5% is coding sequences, and the rest, all percentage of the sequences, which is 98 to 98.5%, is actually junk sequences, and these are the interspersed region in between the DNA. Many of them are introns. So why you have that advantage in the eukaryote? This is an advantage. So you actually hide the genes which actually are essential for maintaining the life into a lot of these junk DNA. So what happens that if by chance a part of the chromosome is broken or damaged during the cell division, most of the genes are actually not damaged. The chances of getting the genes damaged are much less, because this is like hiding a needle in a haystack, and that is not, or that mechanism is not possible without an intron in the bacteria and the archaea. But they are so primitive and they actually so rapidly grow that they really do not have a problem in maintaining or sustaining if there are some breakage in the DNA, because they actually grow much faster. On the other hand, in an eukaryotic organism, if you actually miss a gene, or if you actually do have a gene which is actually damaged after the cell division, then it can have very, very serious, even lethal consequences. Nucleolus is, of course, not present in the archaea and the bacteria, whereas in the eukaryotes, the nucleolus is present, which is actually made up of RNA, nuclear RNAs. And the mitochondria, chloroplast, endoplasmic reticulum, Golgi, lysosome, these are all the organelles which are membrane-bound organelles, are not present in bacteria archaea. Of course, it's present in eukaryotes. And the plasma membrane lipid, this is a big difference, is ester-linked phospholipids, and some also do have sterols. And in gram-negative bacteria, one of the important part is that you have an outer membrane which actually have a lipopolysaccharide. But here we are not talking about the outer membrane. We are talking about the membrane which encloses only the cytoplasm, that is the plasma membrane. And the glycerol diether and glycerol tetraethers are actually present in the archaea, very different. And they also have isoprene, whereas these are the branched lipid parts, and the isoprenes are not present in the bacteria as well as in the eukaryote. Eukaryotic also, we have the ester-linked phospholipids and the sterols like cholesterol that gives the membrane rigidity. That cholesterol is absent in archaea, but isoprene is not mentioned here. Remember that the archaea also have the isoprene, which is a branched lipid that is present in that is absent in bacteria as well as eukaryotes. The flagella is actually submicroscopic in size, and the filament is composed of a single type of flagellin protein. And this flagellin protein is actually deposited at the very top of the flagella in the process of flagella building. And the proton motive force, again repeating, the proton motive force actually drives this flagella when the protons actually fall down from the periplasmic space into the cytoplasm. That proton motive force drives the flagella. That's the energy of the flagella. On the other hand, the submicroscopic in size, of course, and some of the filaments are composed of more than one type of archaeolin proteins. And these are called the flat type proteins, BA, those kind of proteins, more than one type. And also another difference is that these archaeolin proteins, which actually makes the archaea, which is the archaeal flagella, they are actually attached at the base instead of at the top, like in the bacteria. And also the ATP are directly used for the locomotory purpose, or for moving the motor of the flagella or archaea here to do this, not proton motive force. That's a big difference. In eukaryote, they are microscopic in size, and they are actually membrane bound, and usually they're made up of 20 microtubules, about in 9+2 patterns. This means like you have 1, 2, 1, 2, 1, 2. You have 9 of these microtubules in the bunch of 2 arranged in a circle, and in the middle of that you actually do have just only 2 of them. You can just take a look at the textbook, the cross section of this one, just to have an idea that what we mean by this 9+2 pattern. This is a cross section of the flagella. Say for human being, we have only the sperm cells which actually do have a flagella, but there are many types of eukaryotes, protists, which are the protozoa, which actually do have flagella. Many of them actually use that for the swimming purposes, and many of these flagella containing protozoa actually are disease organisms, like say for example trypanosoma. Trypanosoma actually can cause sleeping sickness, that actually do have a flagella. Euglena, which is a photosynthetic protist, that does have flagella. So there are many prokaryotes which are eukaryotic cell do have these flagella. Then the ribosome size and structure, you can see that it is a 70S structure, and they are made up of the 2 subunit, which is the small subunit is 30S, large subunit is 50S, and the subunits are also, the large subunit is made up of 23S and 5S, whereas a small subunit is made up of 16S ribosomal RNA. The rest of it comes from the protein. So on the other hand, the archaeal ribosomes are also 70S. Similar to that, you can have 16S ribosomal RNA in the smaller subunit. Larger subunit have 23S and 5S ribosomal RNA, but in addition, it may have a 5.8S ribosomal RNA as well, and the rest are protein. But the most important difference is that these ribosomal proteins in between the bacteria and the archaea differ so much. That's the reason that they are put in two different domains of life, and the phylogenetic tree was changed based on these sequences. On the other hand, you can see that the eukaryotic ribosome is 80S, and the small subunit is 40S, the large subunit is 60S. They are made up of 4 ribosomal RNAs, and there are more than 80 ribosomal proteins, whereas the number of ribosomal proteins are much less in the bacteria and the archaea. Actually, cytoskeleton is actually very rudimentary in those, but we actually talk about that there are FTSZs and the MREBs, MBLs, and the CRES. And we talked about that there are some analogous proteins in the cytoskeleton. Earlier, scientists used to think that there is no cytoskeleton in the bacteria or the archaea, but they have analogous proteins, and they do have a rudimentary cytoskeleton. Whereas in eukaryote, the cytoskeleton is very, very well developed as well. So the gas vacuoles are present in the bacteria and the archaea for the buoyancy of this bacteria and the archaea, which is not present in the eukaryote. So that's pretty much sums up that what are the differences between the bacteria and the archaea and the eukaryotes. And that's pretty much the approach the material comprehensively, compare, contrast information in between the 3 domains, and look for the similarities and the differences to best understand the overall themes. And that's pretty much it. That's the end of chapter 5, section 5.8. That is the only part that is actually needed.